Greetings, students. In this section of our lessons, we're going to talk about how the cell cycle is controlled, and then we're going to move into talking about meiosis and how the gametes or sex cells are formed. But let's first start where we left off in the previous lesson, which is section 8.7. The rate of cell division is affected by environmental factors. In laboratory cultures, most normal cells divide only when attached to a surface. And what happens is the culture cells will continue to divide until they touch one another. Most animal cells will divide only when stimulated by something called a growth factor, whereas some animal cells don't actually ever divide at all. Okay? Growth factors are a special class of compounds whose job is to stimulate the cells to divide. So, as we mentioned in the previous slide, let's say we were in the lab and we have this dish and we put some cells in it. The cells, once they adhere to the surface, will actually start to grow and divide, and they'll continue to grow and divide until they actually sense themselves contacting each other in some kind of a single layer. And then what will happen is as they start to sense each other, they'll slow down and stop dividing. Now remove a section here, kind of like imagining like you cut your knee or your finger or something here and you have that chunk of skin missing. The cells can actually sense that absence, and that will trigger the cells to start growing and dividing again and increase the speed at which that happens. And as the cells will start to sense each other around and start touching each other in different directions, they'll start to slow down production again, and then you'll end up with this layer of material. Now, most part of this process works quite normally, but there are times when it doesn't work properly, when these cells continue to divide uncontrollably. If that's the case, then we end up with something called a cancer. So if you look at this last slide here, you can see this uncontrolled growth of cells here. The cancer cells are forming clumps of overlapping cells. Now let's define what a growth factor is. A growth factor is a set of proteins within the cell that controls the cell cycle. Remember, the cell cycle is actually detects signals. These signals are able to affect different checkpoints in the cell cycle to determine whether a cell should go on through completing the cell cycle or it should stop. And what will happen is the growth factors will actually bind to special types of receptors on the plasma membrane, and that will allow the cell to detect what's going on within its environment. Remember, we talked about those when we talked about the plasma membrane earlier in the year. So it looks something like this. Here's a particular growth factor. It's just going to direct the cell to do something. It's picked up by a receptor protein, and then that message is sent on to the cell. And if the cell is moving through the cycle, we have some checkpoints here. We have a checkpoint at G1. If it gets a red light, it means stop. If it has the all clear, everything looks good, it will move on into the synthesis phase. And then into G2, there's another checkpoint. And then into the M phase, there's also another checkpoint. So checkpoints along the way, make sure the cell is proceeding properly. If there's issues within the cell itself or within the environment, which might stop the production of the cell, or continue the process of moving into the cell cycle and dividing. Here's an example of how this growth factor affects the division of the cell. If you look in the side here, you can see these cells here have no additional growth factor added to them, so they don't continue to divide. Whereas on the right-hand side, a growth factor has been added. The cells can detect that growth factor, and then that will trigger them to actually start dividing. Now, the cell cycle is regulated by a special class of chemicals called cyclins. And this class of chemicals is actually only discovered in like about the last 40 years. These special types of proteins actually control the process as the cell moves through the cell cycle. And like I said in the previous slide, at G1 there's a checkpoint. If a signal is given, the cell will move on. If no signal is given, the cell will enter what's called G0. And at that point, it will no longer divide. And some types of cells in the human body actually basically stay in that stage, G0. For example, brain cells and muscle cells. So imagine one day scientists are able to do research and make discoveries about how to get some of these types of cells to actually kick into the cell cycle and actually move out of G0 and then in through interphase and then through mitosis. For example, let's say you suffered a traumatic brain injury or a nerve cord injury. Those types of cells and tissues are not going to heal themselves generally as a rule of thumb. But imagine one day if we could actually get those cells to start dividing and growing again, so that way they could actually replace the damaged cells. That would be something very exciting. Some cells do have the ability to actually re-enter the cycle if they need it, for example, liver cells. And here's an example of something that might be done in a lab where you have a cell that's actually moving through the cell cycle. You remove some of that cytoplasm and then inject it into a cell that's in a different stage of the cell cycle. And it actually, the introduction of the cyclin actually will push the cell to actually move into mitosis. Now, when we're talking about cells, there are two different types of regulators that the cells will respond to. There are internal regulators and external regulators. Internal regulators are those type of regulators that the cell will respond to that occur from within the cell. That's why they're called internal. And those particular type of regulators will direct the cell to proceed only when certain events have occurred in the cell itself. External regulators are those types of regulators that are outside of the cell in the external environment that the cell can respond to, like, for example, sensing the cells touching each other so that the cells will speed up or slow down the growth and division of the cells. Then we also have a special case called apoptosis. Apoptosis is the programmed death of a cell. And this is very important, actually, because this process, almost, if you will, like cell suicide, prevents unwanted cells, like an excess amount of cells that you don't really need or want. And it also helps eliminate defective cells. Now, when the cell cycle doesn't run properly and you have cells that don't respond to the cyclins, the growth factors, the regulators that we talked about, that is an example of a cancer. Cancer are cells that divide excessively to form masses called tumors. And there's two classes of tumors. You have benign tumors, which are tumors that grow but they don't invade the surrounding tissue, and then you have malignant tumors, which do. The primary method of treatment today is radiation and chemotherapy, which are used to as cancer treatments, and these kind of target those cells that are dividing very rapidly. And the problem is with these types of treatments, especially like chemotherapy, is because they hone in on those particular type of cells, they have a tendency to also hone in on those types of healthy cells in the body, like, for example, the cells around your hair follicles and stuff. That's why oftentimes one of the side effects of some of these treatments is hair loss. This is an example of a tumor growing within a breast here. You can see that's why it's so significant to have regular checkups for these kind of things and monitoring, especially as you get older, and have these kind of tests done to make sure, because if a tumor is caught early enough, it can oftentimes be removed with, say, surgery. But if it starts to get invasive like this, then it becomes more complicated for the doctor. And then it can actually become to the point where it metastasizes, which is very dangerous because now it enters both the blood system or the lymphatic system, and at which case now it starts to spread to other parts of the body. And at that point, it becomes very difficult for doctors to deal with. All right, so that closes out mitosis. And what we're going to do now is shift gears and talk about meiosis, which, of course, is the cell division that produces the sex cells or the gametes. What we've been talking about so far is the division of the body cells. And we also sometimes refer to body cells as somatic cells. These are the cells of an organism, for example, like us, that has the specific number of chromosomes that you find in that particular type of organism. For example, human cells have 46 chromosomes. Okay, so a body cell will have 46 chromosomes divided up into 23 pairs, homologous pairs of chromosomes, which means two chromosomes together that are basically the same size and shape and contain the same types of information. Now, the chromosome of a homologous pair carries genes, and those genes code for particular types of traits or characteristics, and you'll find those same types of characteristics or traits exhibited on the other chromosome in the same spot. We call that a locus. Now, it may not be coding for the same exact trait. For example, both spots on each corresponding chromosome might be coding for, say, for the way the ear is attached to the side of your head. But each chromosome may have a different code in terms of how it literally is attached. Maybe one is coding for dangling, one is coded for attached. Okay. This is what a homologous pair of chromosomes looks like. You have two chromosomes together. You can see they look like the same in size and shape. The locus is the corresponding spot on each one that's in the same place that contains the same type of genetic information. Although the code may be a little bit different, it's coding for the same basic trait. Now, cells with two sets of homologous chromosomes are said to be diploid. That's like that. Gametes, which are your egg and sperm cells, are haploid, which is something like this, just one chromosome. They have a single set, whereas your somatic body cells have two sets of chromosomes. Organisms that are reproduce sexually, go through a sexual type of life cycle, will involve the alternation of generations, or the alternation between haploid and diploid stages, which means this. Let's very simply have this couple here. The female has the ovary, the male has the testes. The female will produce the egg in the ovary, the male will produce the sperm in the testes. These are the gametes here, or sex cells. Each one is haploid, which means they have half of the genetic information. N represents the number of chromosomes. That's the haploid stage. When they come together and fertilization takes place, now you have a zygote. But that zygote is diploid now. So now it has the full complement of chromosomes. It has 46 chromosomes, which is typical, of course, in a human, or 2N, which is double the number of chromosomes, or 23 pairs. And then from there, the cells will divide, grow, reproduce. Mitosis will take place, and you'll grow up to be an adult like this, and then the cycle will spin around again. Now, meiosis is a little bit like mitosis in that some of the stages have the same names, but it does involve an interphase stage where the chromosomes are duplicated. And then there's two divisions that take place in meiosis, meiosis 1 and meiosis 2. Meiosis is sometimes referred to as a reduction division because as the process of division takes place, it's actually reducing the number of chromosomes. So chromosome duplication only takes place one time. Okay? That way, when the process is all done, each daughter cell will be haploid. Meiosis, like mitosis, which I said in the previous slide, is preceded by a chromosome duplication. But remember, in meiosis, the cell will divide twice. In mitosis, the cell only divides once. So in mitosis, you end up with 2 daughter cells. In meiosis, you will end up with 4 daughter cells. Now, in the first division, which is called meiosis 1, what will happen is, after the genetic information is duplicated, the chromosomes will line up in homologous pairs, side by side. Something like this, which they call a tetrad. And then another thing that's unique to meiosis will occur is something called crossing over, in which these homologous pairs will actually exchange genetic information. But we're going to talk about that in our next lesson. Now, what will happen through the course of meiosis 1 is you'll go through the same named stages that we learned about, prophase 1, metaphase 1, anaphase 1, telophase 1, similar to mitosis. But what's going to happen at the end of meiosis 1 is the homologous pair of chromosomes will separate. So each daughter cell will have one set of chromosomes. So the 2 chromosomes will become 1. Each daughter cell will get 1. And then you'll roll into meiosis 2. And in meiosis 2, the steps are, again, prophase, metaphase, anaphase, telophase. Both would be prophase 2, metaphase 2, anaphase 2, telophase 2. And in this process, it's almost the same as mitosis. Now, between meiosis 1 and meiosis 2, there is no duplication of the genetic information. So after the daughter cells from meiosis 1 divide, those chromosomes will now be passed on to their daughter cells, but each daughter cell will have half of the genetic information because it did not duplicate. So in the end, each of the cells will get some of the sister chromatids, and you'll end up with 4 daughter cells that are haploid. So what does that look like here? So we have interphase. Here's where the genetic information is duplicated. We roll into meiosis 1. In meiosis 1, the big thing that's going to occur is crossing over, which is the exchanging of the genetic information within the tetrad, which we'll look at in the next lesson. Then you have, in metaphase 1, the chromosomes lining up and has homologous pairs in the center, or the equatorial plate there. Notice they're in a pair side by side together. And at that point, what will happen in anaphase and telophase is the chromosomes will separate, not the sister chromatids. See, it says they remain attached. What's actually separating, literally, is the chromosomes. Okay, it's like that, this chromosome. So the chromosomes are separating apart. And then in telophase 1 and cytokinesis, those cells will continue and finish dividing. So you end up with 2 daughter cells, and they each have their own corresponding genetic information. Once we move into meiosis 2, there's no duplication of genetic information here. So you can see here, this daughter cell and this daughter cell will go through the process of meiosis 2, which is quite similar to, like we said previously, mitosis, in which the genetic information will line up in the middle. And then in anaphase 2, you'll see now the chromatids separate. So now the chromosomes are being torn in half. And so what you end up with at the end is 4 daughter cells, each one haploid. Each one has half of the genetic information that the parent cell had way back in the beginning, rolling through the process. Now, both mitosis and meiosis begin with diploid parent cells that have had their chromosomes duplicated. But mitosis will produce 2 genetically identical diploid body cells or somatic daughter cells, whereas meiosis will produce 4 genetically unique haploid gametes or sex cells. They'll be different, and they'll have half of the genetic information. Why they're unique and different, that's going to be our next lesson. So the process looks kind of something like this. If you want to compare and contrast them, each one is a cellular division, and the stages are the same in the names, sort of. We have prophase here and prophase 1. You can see some differences here. You have the duplicated chromosomes in both. But in this one here, you have the homologous chromosomes coming together in pairs. They don't do that in mitosis. And then you also have crossing over taking place. In metaphase, you'll see the chromosomes line up. In metaphase 1, the chromosome pairs line up. And in anaphase and telophase, the cells will divide. But in this case here, the chromatids divide. Here, the chromosomes divide. And then when you run into meiosis 2, that's where the process looks a little bit more like this, ultimately with the chromatids separating as the end result. All right, so that's where we'll stop for today, and then our next lesson we'll finish up talking about meiosis and then move into the last part of the chapter.